Thank you all for coming out. We're really excited to host this event for the region today. I think it's going to be a fantastic discussion, really interesting topic. We're excited to kick off this new event series of ours, the CEO Talks, to complement the general networking events that we do each and every month. Of course, our mission is to build a more connected workforce in the region, and that workforce is everything from CEOs and entrepreneurs down to the bench. And there couldn't be a better place for us to kick this event series off. We've done a couple events here at Montgomery College, and our sponsor, PICMC, the Life Science Innovation Center here at the college, they host startups and businesses. So this is a community college with an incubator accelerator right here on campus. So it's a pretty unique community college, the only community college in the whole country that has a health system right here on campus. So you have a community college where we're teaching the next generation. You have a health system right here. We also have world-class laboratories right here on campus. And it's amazing. I mean, it's pharmaceutical-grade laboratories. One of our sponsors, BioTrack, is here today. Right now, they have a epigenetics workshop where they have scientists from all around the world who are traveling here into Maryland and Montgomery College for these courses. In fact, last week, they had a gene editing with CRISPR-Cas9 course as well, where they had 20 scientists from around the world. So when you put all these factors together, I mean, that's a pretty amazing story, and it's pretty amazing that this is all in one campus right here in Maryland. What's great also is it's a good representation of what Maryland and the BioHealth Capital Region has. That type of activity is happening across the state, across the region. And we have 70 federal labs, we have the best universities, we have a very vibrant biotech ecosystem. So that example of what's happening right here on campus is happening all across the region. So what we want to do, and what we're so happy to have WSGR here as a sponsor, what we want to do is just continue that conversation and make sure everyone knows about that and provide opportunities for you all to get together around topics, create those collisions to make more things happen and bring more products to market. So we're excited to be here. There's a lot of organizations that are involved in getting this event together. WSGR is our lead sponsor, as well as PICMC, BioBuzz, and BioTrack. So we thank them for all their efforts to do so. And there's a lot of organizations involved in this biotech ecosystem. BioHealth Innovation, Rich is here from that. TEDCO, the Maryland Tech Council. So it's really a collaborative effort. And when you look at our region compared to others, we may not have a lot of the same amount of density, but we can out-collaborate. And I think that's a really good mission for us, is we're going to out-collaborate them. And that's what we're starting to do here today. So with that, I'm happy to introduce our moderator today, the very wise Charles Andrus from WSGR. Charlie has an extremely impressive track record. He's got over 70 publications, 4 drug approvals, and but practices not only FDA law, but patent law. So he brings a wealth of knowledge and experience to this topic, and is really going to lead a really fantastic panel with our panelists today. So I'm going to stop talking and give the mic up to you for the majority of our program. So thank you very much.
Thank you, Chris, for selecting this just incredible venue, and for all your hard work and the logistics for putting all this together. And I appreciate the intro because it creates no pressure, so that's good for me. And we're very lucky, I think, to have 3 entrepreneurs with us today who have companies that will hopefully go on to change the world. They are dealing in whole or in part in the immuno-oncology space. This is, I think, in my opinion, sort of one of the 3 future areas of medicine that are going to have significant impact, the other 2 being regenerative medicine and epigenetics. They each have, their companies are each taking a different approach, which is interesting. They're represented geographically in different places. For example, Paul and his company are located on the West Coast. And they also have different levels of entrepreneurial experience. Jeff, for example, is a seasoned entrepreneur, whereas Paul is a little bit earlier and still kind of earning his stripes.
Nice way to call me old. I saw that.
I got more gray hair.
So I think it would be appropriate, before we get into the questions, maybe if each of you could introduce yourselves and maybe tell the audience a little bit about your companies.
Okay,
Okay,
I'll go
Bill,
first.
you're up first.
So I'm Bill Hurl. I'm the CEO of Immunomic Therapeutics. Immunomic Therapeutics is about 10 years old. We were founded on a technology out of Johns Hopkins, and we took a different route to immuno-oncology than a lot of people might have suspected. We started out actually in the allergy area, and we picked allergy as sort of a low-cost option for us to prove that our technology works, with the idea that we would come back to immuno-oncology once we were successful with the allergy proof of concept. And very rarely do you actually have a business plan that actually does what you thought it was going to do. But in this particular case, everything kind of played out perfectly, and we ended up with a very large deal with Astellas Pharma that took the whole allergy business and left us with a nice little nest egg to come back and work in immuno-oncology. So our relationship with academic collaborators allowed us to really jump right into a program for glioblastoma, which is now in phase 2 at University of Florida and at Duke. And so it's a very promising cell therapy for glioblastoma, and it's something I'm very excited about. So like I said, we took a different route than a lot of people. We never had any VC funding. And so if you can imagine a 10-year-old biotech company that actually turned a profit with no VC funding, that's a little bit different than many. And so it's a different strategy. So it's been an interesting ride. I have many curious and fascinating stories to tell, I'm sure. If you want to take some time to talk about that.
Bill, maybe we could just clarify that, because I think that was a bit of an understatement. You said you had a little nest egg. Could you maybe tell the audience just how little that nest egg is?
Well, yeah, that was, we did a $300 million deal with Astellas. So that was $300 million in cash. That was not BioBuzz. That was real money. And so I have to tell you that one of the best days of my life was when my CFO came in and said, hey, look at the Bank of America balance. And the number on the screen was like this. And I was like, yes.
Okay, so for those of you who want to put aside a little nest egg, you have something to shoot for. Thank you, Bill.
Yes.
Jeff, maybe you could go next.
Sure. Hard to follow that one. We don't have a nest egg, but that sounds like an inspirational story. I'm going to keep that in mind. Put that in the business plan. So our company. So I'm Jeff Galvin, CEO of American Gene Technologies, and our company is also 10 years old. Started when I met a guy at National Institutes of Health who showed me viral vectors. I come out of the computer space, and looking at viral vectors and knowing what I knew about the technology and how microcomputers revolutionized the whole market, and and looking at the cell now as an organic computer and our ability to actually change things in it to mitigate disease, my head practically exploded. I just said to him, somebody's got to be the Microsoft of this business. Might as well be us. And he didn't know what I was talking about, but that was really my vision for the company. It was that delivering the goods was going to be a critical component in every application that eventually gets written for the human computer. And that if you focused on a platform of tools and enabling technologies that allowed you to make these applications more quickly than anybody else, that you'd be a go-to collaborator in this space over the long term. In other words, build it and they will come. Well, that's turned out to be true. So, of course, nobody buys platforms, they buy products. That's just common wisdom in the industry. So at some point when I started to sense that the financial markets were ready for this, we decided, okay, let's take our lead developments and propel them into the clinic. And we also have no VC funding. All of our money came from high net worth individuals, some angel funds, and some boutique investment companies. And we were able to find a fairly inexpensive way to get our HIV functional cure into phase 1 trials. We expect that to happen this summer, and we believe we have a functional cure for HIV, that we can restore natural immunity to HIV by doing something that looks a lot like CAR T. We pull out T cells, modify them, put them back in. We restore your ability to fight off HIV instead of having your HIV-specific T cells depleted or co-opted by the disease. Instead, you fight it like you fight the flu and the cold, and once you have memory effect of those T cells, you're immune for life. You'll mop up your own viral reservoir, and you won't have to take antiretroviral therapy. And I got to say that everything I'm seeing in the preclinical data at this point is confidence building. And I'm not a molecular biologist. Like I said, I came from the computer space. But when we show this to people that really know HIV, you can see it, the excitement and sort of the belief that they have in this as well. So, but that's just the start. I mean, the idea is prove the technology and then create a machine that turns out these drug apps that are de-risked for pharma. We feel like we can hit a good inflection point. That's why I was like, inspirational story, right? If we can go ahead and do something that's really good for Roche or Pfizer or Gilead, I mean, you can imagine that HIV cure would be of interest to Gilead. We could go ahead and bank some cash that we can use to just drive this machine that's really my dream, which is to action gene and cell therapy for the broad audience, for the public, as quickly as possible. So we have phenylketonuria right on the tail. That's a monogenic disease. And then we have Immuno-Oncology, where we have a vector that stimulates the primary tumor in any epithelial cancer, causes the gamma delta T cells to activate and start eating at 300 to 600 times their normal level in the microenvironment. We're getting 75 to 85% complete remission in mouse models, and abscopal effect. So these circulating gamma delta T cells, once they're excited, they tend to clean up the rest of the epithelial cancers in the mouse. It doesn't even have to match the primary tumor, because gamma delta cells, and you're going to hear a lot about gamma delta T cells over this next year, they exquisitely select for their targets. They've never been associated with an autoimmune disease. They're really good at eating cancers. The trick is how do you get them looking at the cancers? It could be, remove a checkpoint molecule. It could be, doing something to the gamma delta T cells. We're seeing that actually Bluebird is planning to put cars on gamma delta T cells. Our philosophy is treat the cancer, not the immune system. Immune system's complicated. If you can leave it in its natural state, that's the safest way to go. And we think we have an approach where one vector to the primary tumor could even work in a late stage cancer, and we're starting to gather a lot of data that should be the case. That'll be a 2020 clinical trial for us, we believe.
Thank you.
Thanks.
So-
Sorry for going so long.
No, it's okay. I think you brought up an interesting point there, right? So initially, it kind of sounds like you're moving along something that seems to be an emerging paradigm, which is initially you're just looking for something that provides some kind of therapy.
Mm-hmm.
And then the idea is eventually to take a disease which can be terminal and convert it to something that you can treat chronically. And then, the next step in that is actually to, quote unquote, cure the disease. It seems like you're kind of moving along that pathway, maybe with the goal of transitioning from chronic treatment to an actual cure.
Yeah, I see how that could be a takeaway. So let me tell you what my thinking really is, right? So I come out of software, and my whole life in software has always been the same. It's you have this limited tool set, and you're trying to create value. That's it. And so it doesn't matter what the low-hanging fruit is. You can't survive as a software company unless you can figure out how to creatively combine things that already exist, whether you develop them or somebody else, and come up with something that has some value to the customer, right? So customers are complex in the healthcare industry, but basically we just focus on the patient. If we have something that solves a patient problem, we think that will eventually find its way out to those patients. So remarkably, a complex disease like HIV became the low-hanging fruit, and a lot of it was just serendipity. you know, that the right team gelled around it at the right time, and just coincidental things that I thought of in, you know, while taking a shower seemed to get some traction with people that actually knew HIV. And I was just thinking about the tool set, and all of a sudden something's there that we could action. But we don't care what it is. We don't care whether it's a treatment, a cure, a not, you know, something that will knock back a disease for a period of time, and we can start to treat it chronically. We just think, how can we move the needle in efficacy in healthcare? That's the critical thing that we're just keeping an eye on. And I was just saying this to Paul earlier, I haven't met a company in this business yet that I didn't think had tremendous potential, because it's so big. You know, there's 6,000 monogenic diseases that are completely untreatable right now, but the palliative care is $2 to $4 trillion a year in the healthcare system. How would you like to move that to pharmaceuticals, right? Here you go, this panel's doing it, right? And that is the potential. It's not, you know, there'll be more than, he's got, you know, several $300 million payouts in his future, I guarantee it. Hopefully all of us on this panel have that. That is the land grab that is gene and cell therapy today.
And I think it's a good point, right? Before I took on my second career as a lawyer, I spent 10 years working at a no-name pharmaceutical company called Bristol Myers Squibb. And at the time, the model that Bristol operated on was blockbuster. Everything had to be a blockbuster drug. And in 2020, Bristol Myers Squibb is slated to become the #1 orphan drug seller in the US. And so people are looking at things like the Orphan Drug Act. They're looking at these unmet diseases. You mentioned 6,000. I think the number in some estimates is closer to 8 or 9. And each one of those represents an opportunity, because many of them have only sort of palliative type treatment, as you were pointing out. So thank you.
Sure.
Paul.
So my name is Paul Toomey. I'm a co-founder and CEO of BioGraph 55. We're a little less than 2 years old. The two other co-founders are Tony Rebos from UCLA Medical Center and Peter Thompson from OrbiMed. And we founded BioGraph 55 to help solve some of the biggest challenges in immuno-oncology today. And that's that despite clinical success, most patients do not respond to single-agent checkpoint inhibitors. And so we developed a technology platform at UCLA. We hand-carried that technology platform over to the biotech side. And it's a target discovery technology platform. And so with that, we've identified targets that we believe are differentiated. And our therapeutic strategy is focused on not converting tumors from T-cell non-inflamed to T-cell inflamed. Our therapeutic strategy is to convert T-cell inflamed tumors that are resistant to T-cell inflamed and responsive, because from the work we've done, we believe that T-cell inflammation is required but insufficient to achieve a clinical response. And so we are currently in our seed stage of financing. The last 2 years, actually, we very quickly built out the platform. It's been validated on a technical and clinical level with over 10 different partners, 5 of which are coming back. And during that time, we made these internal discoveries that have led to us evolving from sponsored research towards to developing drugs. And so the goal is we're about to close our seed stage financing, and we anticipate that we would raise and close our Series A sometime towards the end of 2018.
Thank you. So that raises a couple of questions in my mind. The first is, you're talking about, I think, really the cancer environment itself. And people will generally say that cancer is sort of a random thing. It happens to you because you were out in the sun for too long, or you pumped too much gas, or you ate too many foods with like weird additives that haven't been in nature before. You know, what are your thoughts on cancer and randomness?
I don't think that there is anything random about cancer. In fact, with the, you know, my focus is the melanoma tumor microenvironment. It has grown to multiple solid tumor types, but I think it's, what Charlie's raising is a very important point, that cancers are not a randomly built set of networks. And we know very little about how these cells within the tumor communicate with each other to promote their survival. And what we're finding is that there are patterns that are conserved in subgroups of tumors. And so if there is a pattern, by definition it is regular, which means that there is an underlying mathematical structure to that pattern. So what we've done is we've applied graph theory to understanding how are cells talking to each other? Where are they? What are the underlying cellular networks that lead to emerging properties for these cancers to be able to grow in a host? Because we've been designed, you know, to kill cancer, right? So in order for a cancer to become metastatic and be able to live in a host and a patient, it must have acquired so many capabilities, right? And in order to do that, it's got to communicate with host cells. So what we're finding is that that pathologic communication is actually very conserved. It's an important point because if you want to select the right patients for a particular treatment, right? We are, you know, the clinic is in urgent need of combinations that work, right? And we need to know which patients are most likely to benefit. Well, how can you do that if you don't share any genomic overlap? Meaning, if one patient has a particular mutation landscape, right, and another patient has a completely different one, well, how do you select those patients for the same treatment, right? If you go up one level of biological organization to the cellular networks, we're actually finding that patients that have a particular cell-to-cell communication sitting in one particular location of the tumor are much more likely to respond to treatment, to say a single agent anti-PD-1, than others. So our view of cancer is that it's no different than your normal organs, except it's got cellular networks that are pathologic.
Okay. So, you know, you sort of also, I think, hinted, and I'm going to throw this out to Bill,